guys want to actually play it?
I'm sorry, what did you say?
Would you rather do the live quiz, or do you want to just go through the questions one by one?
I'm fine with whatever.
Okay. All right. Let's just go through the questions one by one. That way we can take more time and explain them if we need to. Okay. So, I mean, you guys just shout out the answers, type them in the chat, whatever you're more comfortable with. Let's try and make this interactive. That way we can, I think that'll just help us more. So all cells are surrounded by a thin, flexible barrier called a
Cell membrane.
Yep, cell membrane. Exactly. So cell wall only applies to plant cells, right? A photocell, I'm not really sure what that is. An electron microscopy is a technique. Okay. Blank are cells that enclose their DNA in nuclei. What would that be? Well, two of these I've never heard of. Is it prokaryote or eukaryote? So these obviously aren't AP questions. These are just to get our get the ball rolling a little bit. Eukaryote. Yep. Good. Next question. Megan has a large membrane enclosed structure within her cells that contain her DNA, known as a nucleus, right?
Yeah.
Okay. Yeah, if you're comfortable, Mira, you can just shout them out. That way we can just go through these really quickly. In general, eukaryotic cells, including plant and animal cells, are blank than prokaryotic cells. They are
more complex.
Yep, more complex. What is the total magnification of a microscope with an eyepiece magnification of 10 times and an objective lens magnification of 50 times? So I'm not really sure, but I would think 500, right? What do you guys think?
That's what I would assume.
Okay.
Makes the most sense.
Yeah, it would make sense because it's 10 times and then you multiply that by 50. Okay. Blank are located near the nucleus, help organize cell division, and are not found in plant cells. So which of these are not in plant cells? We actually recently learned about this. Hiba, do you want to try this one?
Centrioles?
Yep, centrioles, exactly. Because, so a centriole is a type of MTOC. It's a type of microtubule organizing center. And remember, plant cells don't have centrioles, but they do have some sort of other type of MTOC. Okay. Blank cells contain an internal membrane system known as the blank, where lipid components of the cell membrane are assembled along with proteins and other materials. Okay. Do we know what this is?
The first eukaryotic and endoplasmic reticulum.
Yes. It's actually, it should be the endomembrane system. But the ER is where the lipids are formed, actually. But yes, the system itself is called the endomembrane system. That's something you should know. Okay, good. Alan, we're just going through some quick factual recall questions, and then we'll go through some AP questions, all right? Blank are the biological equivalent of a solar cell. We learned about this when we were dealing with photosynthesis and when we were dealing with cell respiration. So just as review, what does the mitochondria do? Which of those two processes is it involved with? The mitochondria, powerhouse of the cell.
Cellular respiration.
Exactly, cellular respiration. So chloroplast is the answer, which Mira said. Okay, and that's responsible for photosynthesis. Okay. Most prokaryotic cells produce a strong supporting layer around the membrane known as, you guys know this one? Nope. Okay. It's just a cell wall. So we learned that plant cells have a cell wall, but it actually happens that that bacteria also have a cell wall, and they have what's called peptidoglycan in their cell walls. All right, bacteria, and then it can be either gram positive or gram negative, and that's something we'll learn about later. But just keep that in mind. All right. Yes. So bacteria have, and archaea, of course, have cell walls. Okay. All right. Good. Whoops. Okay. There's 20 questions, and we were on. I think yes. Okay. Sorry. The blank regulates what enters and leaves the cell and also protects and supports the cell membrane. Exactly. And why is the cell membrane good at regulating? This is one of the FRQs, I think. Because it's semi-permeable. And why is it semi-permeable? Alan, you can shout it out or you can type in the chat. Okay, good. Phospholipids. I know, Mira, you're typing. So, and why are phospholipids good at regulating? So that's really how theory, exactly, because they have a lipid region, which is non-polar, and they have phosphate heads, which are polar. So it's good at regulating. It has both characteristics. So that's really how thorough you want to be in your explanations in FRQs. Okay. Particles moving from a high concentration area to a low area of low concentration are demonstrating what? Diffusion. Good. Now, this is something that always gets people. When you have diffusion of water, it's called osmosis, right? If you have particles, but when does osmosis happen? I think this is something that confuses people, is if you have osmosis, if I have, yes, osmosis happens with water. But if I have water and I have, let's say that, let me draw here. Can I not hit Control Z? Oh, I know why. Because my thing isn't plugged in. That's why my keyboard wasn't working. Okay, all right. So let's say we have this semi-permeable membrane that looks like this, and these, I have 3 big sugar molecules here and just 1 big sugar molecule here. This is what type of solution? Is this hypertonic, hypotonic, or isotonic? On the left. On the right, or sorry, on the left, it should be hyper, right? Because it has more solute. It's hyper, it's packed with solute, right? That's how you want to think about it. It's very hyper, right? Exactly. Hyper, and then the right, obviously, then would be hypo. So water, how do you think water would move? Obviously, the reason why the particles can't move from an area, the first thing that the particles would try to do would be to move from this area of high to low concentration. But they can't, because these holes, as you can see, they're too small. The particles can't move through them. So water is forced to move. So how would water move? Would water move from hyper to hypo or from hypo to hyper?
Hyper to hypo.
And that's the problem, right? We think that water would move from hyper to hypo, but think about it. What does that do actually? That makes the problem worse, right? Because I'm jam-packed with solute on the left right now, and I have lots of water on the right. If I keep adding more water to the right, then I'm going to be even more jam-packed with solute, and I'm not going to solve the problem. So think about it in terms of how do I solve the problem? And I think that's a better way to think about it, because we think it's high to low concentration. But the way we should be thinking about it is when water is moving, we're concerned about the concentration of water. So water is moving from high concentration of water to low concentration of water. That's one way you can think about it. But that's how we should be thinking about it. Yes, so the solvent, exactly. And so usually in biological systems, our solvent is water. So that's what you want to think about. When you have osmosis, it's the solvent's concentration that's important. So and if these holes were big enough, right? Let's say that these holes weren't this small, and let's say the holes were here. Let me redraw my semi-permeable membrane. Let's say these were the holes. These holes are decently big enough. I think that the particles would be able to pass through. Then water isn't really moving back and forth. I mean, of course, it's organically moving back and forth, but the net movement of water is 0. The solutes overall will, yes, water is in equilibrium because water is not concerned, right? Water is not being affected. The problem right now is the solutes. The solutes can easily pass through. Osmosis becomes an issue when the membrane, the holes in the membrane are too small. I think that's a good way to think about it. So now this is diffusion, exactly. This would be passive or active diffusion, what we're doing right now, what I just showed. Passive, right? Because it's going from high to low concentration without any energy. And this would be, sorry, of course, it's diffusion. Diffusion by definition is passive transport. I'm sorry, I made that confusing. Passive transport is high to low, active transport is low to high, and passive transport, another name for that is just diffusion, okay? So this is diffusion. Okay, all right. That was a good review of that entire chapter, I think. Okay, molecules that are unable to diffuse across the membrane may use blank or special protein channels to pass through. Facilitated diffusion, right? So remember, so protein, just because there's a protein in the membrane doesn't automatically mean that it's active diffusion. The only time you should assume active diffusion is if you see that little lightning looking thingy with ATP inside, because it means it uses energy. If not, if you see a protein and you see a bunch of particles on this side and you see like 2 particles on this side, this going through, that's facilitated diffusion. It's the protein is facilitating, it's making it easier for these particles to go through. Okay. All right, so should be facilitated. Good. All right, next question. Isotonic, as pertains to water and sugar, refers to, this is a weird way of writing it. I wouldn't, it's same strength would be the answer. I wouldn't write it that way. I would say same concentration, because tonicity refers to concentration. Okay. All right. When it is said that the more concentrated sugar solution is above strength or above concentration, I don't like this word strength. It's referring to hypertonic. Yep, because like we showed, more tonicity. Okay. The blank blank gives cell membranes a flexible structure. What gives cell membranes a flexible structure? Lipid bilayer. Good. And once again, we talked about how that's a fluid mosaic model, right? If you kind of put a protein in there, it kind of reforms. It's kind of like what was the analogy that we use in class? Kind of like a salad dressing or something like that. And remember, there was an experiment that was done to show that, right? They I think what it was called freeze fracturing, right? How they freeze fractured it, and then they cut the cell membrane down the middle, and then they showed that there were like little proteins embedded in that. Do you guys remember that? Kind of, sort of. Okay. You just need to understand the concept behind that. That by freeze fracturing, you can kind of break it open. I don't think you need to know all the full details behind that, but yeah. Okay. When the membrane allows one substance to pass through but not another, it's said to be selectively permeable. Exactly, Mira. Good. Okay. The blank converts chemical energy stored in food to compounds the cells can use. The mitochondrion, exactly. Okay, so now would be a good time to talk about cell respiration. A very quick review. Does someone want to try? Alan, Zachary, Hiba, do you guys want to try? I know, Mira, you're on the chat, so it might be a little bit more difficult to type it all out. But do you want to give us a quick recap of cell respiration?
Mitochondria?
Yes, or do you want to give us a recap of the process of cell respiration? You can try, and then wherever you get stuck, I can definitely jump in. There's, yeah, no pressure, just openly, just whatever you can remember.
I mean, I know it's like glycolysis, then Krebs cycle, and then the electron transport chain.
Yep, that's pretty much. And okay, so what happens in glycolysis?
Oh, what is it?
Think about the words. The words in biology really help you. Glycolysis. What is glyco? Glyco is glucose, sugar, and lyse means to transform. What does it mean to cut something up, to break it down? So glycolysis is cutting down glucose, like Mira said in the chat, into 2-pyruvate into water, and it's producing 2 ATP net. And then those 2 pyruvate, like Alan said, go into the Krebs cycle. And do we remember what they produce from the Krebs cycle that goes into the ETC? Anyone? They start with N and F. So the pyruvate goes into the Krebs cycle. What does the Krebs cycle produce that goes into the ETC? Yep, NAD and FAD, NADH and FADH2. Exactly, NAD and FAD. And then the NAD and FAD are they're high energy molecules, they're electron carriers. So they kind of pass down the electrons, and that leads to this electron transport chain, and this little bit of energy is released. And as that little bits of chunks of energy are released, that pumps protons out into the intermembrane space. And as the protons, that creates a proton gradient. As the protons come back in through ATP synthase, it turns that little motor, and motor in parentheses, of course, and that produces ATP, because that motor is essentially phosphorylating ADP. And it's phosphorylating, remember? It's phosphorylating that ADP to produce ATP. And the other thing you need to remember from cell respiration is that the final electron acceptor is, you guys remember what it was? Final electron acceptor?
Oxygen.
Oxygen, good, exactly. All right, that was a quick recap of cell respiration, pretty much everything you need to know. You also need to know that there's 36 through 38 ATP made overall. Okay. Relatively stable internal and external conditions is referred to as homeostasis, right? And okay, there are two ways in which we maintain homeostasis. Do you guys know what those are? Well, cell membrane, yeah, cell membrane is, that's not what I was going for, but you're right. Cell membrane is definitely helps maintain homeostasis, but they're mechanisms. Do you guys remember feedback loops, negative feedback and positive feedback? Yes, of course, I can definitely explain that. Negative feedback seeks to reverse, it's negative, it seeks to reverse what it caused, whereas positive feedback amplifies the response. It makes it more and more positive. So remember how Mr. Rash used that example last year in world history of the triangular trade system and how it was a positive feedback loop? What it did was it amplified the economy of Europe without necessarily benefiting the colonies. So it was a positive feedback loop where it kind of just came back to Europe, and back to Europe, and more and more and more for Europe, right? That's a positive feedback loop. So in the body, you can think about a classic example of negative feedback is insulin, right? When you eat and your body breaks the starch down or whatever you're eating down into glucose, let's say this is a carb, and it's breaking it down into glucose, there's a bunch of processes that happen in between, and I don't have those down on the top of my head, but what will happen is insulin will be released to break this glucose down to maintain homeostasis. If too much glucose builds up, then that's a problem because that gets converted into ketones, and that's a problem. So insulin is kind of, your body's releasing insulin to reverse the effects of glucose, okay? So, whereas if you have, because if you have too much glucose, then insulin production will increase to break it down. Whereas if you have too little glucose, then your you don't want too much insulin. I think I might have reversed that. Please double check that. Maybe I got that wrong. I don't know. But that's the idea, is it reverses the effect. Serotonin, I'm not exactly sure. I know it has to do with, it's like the happy chemical, and I'm not exactly sure the mechanism, but I would think it would be positive feedback. I think one way, positive feedback examples are hard to think of, but let's see. Okay, say for example, I think that's what serotonin does. So like, say I eat some sweet food, right? And I really like it. My body's gonna send some chemicals saying, eat more of that. And then I eat more of that. And then I like that even more. So then my body says, eat even more of that. That's a positive feedback loop. That's what Europe was doing, right? That it's amplifying the response. Okay. Almost done with this one. A group of similar cells that perform a particular function is tissue. Good. And then tissues make up organs, organs make up organ systems make up organisms. And then organisms make up, and we'll learn this later in the year, but organisms make up populations, populations make up populations of the same type all over the world. It's called a species. A population with its other types of, many different types of populations in one area is called a community. Community with all the biotic and abiotic factors is called the ecosystem. Okay, all right. Many groups of tissue work together as an organ. I didn't even know that was a question, but yes, we answered that. Okay, all right, that's enough for the quizzes. Now let's try and do this one. These are some AP questions from the 2013 exam. You can actually probably search this up, the 2013 exam up online and get the whole exam, but Chandana went through and picked the ones that we've learned so far. So we're gonna go through these today, and let me open up the answer key on my end just so I can take a look at that if we need to. And she went through and wrote explanations in as well. And I will share this document with everyone in the Slack afterwards. All right. Okay. So the first question is, let me make this a little bit bigger so that we can all see. How many questions do we have here? I think about, okay, plenty. Actually, I wanted to answer, I don't want to waste too much time. So I wanted to do a quick review of meiosis for Hiba because she asked for that, and then I'll go back to the AP questions. Is that all right with everyone? All right, okay. So mitosis, I'm going to stop screen sharing here, and then I'm going to screen share a whiteboard. Okay. So when it comes to mitosis, we have PMAT, right? Are you comfortable with that, Hiba? Let me see the chat. Yes, okay. So just as a quick review, because if you understand this, meiosis becomes very easy. Prophase is when the chromosomes condense into their form that looks like this, right? Because, and this is a sister chromatid, this is another sister chromatid, and they're connected at the centromere, right? Really, chromosomes actually, they look like this when they're not duplicated. It's in the S phase when they start looking like this, like the X form that we're so familiar with, okay? So this means it's just duplicated. There's 46 of these because we're just dealing with normal body cells, somatic cells, which have 46 chromosomes, which means that this is diploid, right? So in mitosis, we're not worried about homologous chromosomes or anything. In mitosis, in prophase, the nuclear envelope starts breaking down and the spindle fiber starts forming and our chromosomes start condensing. Then in metaphase, they line up like I've shown here. In anaphase, oh, and then I forgot to draw our centrioles, which are in 90 degrees to each other. They pull on this kinetochore right here. They pull on the kinetochores in the middle of the cell, and then they pull that away in anaphase, away for anaphase, and then in telophase, which coincides with cytokinesis, the cell membrane restarts to form. In animal cells, we remember that we have what's called a cleavage furrow, right? And it kind of pinches inwards, and that's the cleavage furrow. And in animal cells, or animal cells, we have a cleavage furrow. And then in plant cells, we have a cell plate that forms, and then that separates, okay? So that's PMAT, very simple. Meiosis is pretty much the same, but we have PMAT 1. Oh, we have PMAT 1, and then we have PMAT 2, okay? Because we have meiosis 1 and meiosis 2. What happens is this is, your body has what are called germ cells. Germ cells are diploid. They are, germ cells are the cells that are responsible for becoming your gametes, okay? So your body has germ cells, and during intercourse, sexual intercourse, the germ cells, which are diploid, become haploid. They become N and N, and of course, this process is meiosis 1. And meiosis 1 is thus called reductional division. It's a reductional division because we're reducing the number of chromosomes. And I'll show you the mechanism by which that happens. And then we have meiosis 2, which is called equational division. Meiosis 2 is called equational division because we end up with 4 daughter cells that have the same, so they become haploid and they stay haploid in meiosis 2. So overall, the process is still called reductional division because you're still reducing the number of chromosomes, but meiosis 1 is called reductional division and meiosis 2 is called equational division. Okay, so once again, in prophase 1, your chromosomes start condensing, okay? But this time in meiosis in metaphase 1, what happens is your homologous chromosomes line up. Because what happens is, remember how we said that in mitosis, it doesn't matter whether they're homologous or not, they just all line up down the middle. In meiosis, however, your homologous chromosomes will line up. And this is called a tetrad. But first, what happens in prophase is there's some crossing over that occurs. So they kind of swap genes and they kind of do this hanky-panky dance where they kind of switch their hand genes back and forth. In mitosis 1, I keep saying mitosis, metaphase 1, they line up in the middle, right? The homologous chromosomes line up in the middle. Does that make sense, Hiba, so far? One of these is from your mom, one of these is from your dad. All right, so these homologous chromosomes line up in the middle. Then in anaphase 1, these are pulled away, and this pulling away, and this is called independent assortment. We'll learn about this in genetics next semester, but this is what creates genetic variation. One is the crossing over, another is independent assortment. That actually comes in meiosis 2. It's not here yet. This is just the separation of homologous chromosomes. The crossing over is what causes genetic variation. And then in anaphase, obviously, they separate, the homologous chromosomes separate. In mitosis, we remember that it was the sister chromatids that separated, right? But this time, it's the entire chromosome itself, the duplicated chromosome that is separating, okay? So then in telophase, we essentially reform 2 cells, but this time, these 2 cells are haploid. So instead of having 2 copies, one from the mom, one from the dad, it has 1 copy of each. Then in meiosis 2, what happens is same thing happens again. Nuclear envelope breaks down. This time there's no crossing over because there are no homologous chromosomes. But this time, of course, we have our chromosomes lined up in metaphase. Then in anaphase, the sister chromatids move away. So as you can see, meiosis 2 looks very similar to mitosis. This is very similar to mitosis. Because as you can see, the number of chromosomes stays the same, and just like in anaphase, we move away sister chromatids. And that leads to, once again, telophase and cytokinesis. We know how that works. And that's why we have N. Okay? And with meiosis, we know that we produce 4 genetically diverse or variant cells. And the reason why this leads to genetic diversity is because this is called independent assortment, because there's a bunch of different chance. Think about the chance that this chromosome could have landed here, this chromosome could have landed here, and then this sister chromatid could have went to this cell, this sister chromatid could have went to this cell. There's just so many possibilities, and we call that independent assortment because it's not dependent on another cell. It's not dependent on another chromosome. So there's all these different possibilities that are occurring as independent events, and that leads to genetic diversity. Okay. Does that make sense, Hiba? I know I went pretty fast, but that's pretty much everything you need to know about meiosis. If you understand mitosis, it makes a lot of sense. Good. Okay. So we've went through about 30 minutes. I'll try and do the best I can to go through the AP questions quickly, but I want to make sure that everyone understands it. Okay. Okay, do you guys have any other questions before we start the AP questions, like any general questions? Okay, good. All right, so let me zoom this in so you guys can see. All right, can you guys see this clearly? I don't want you guys to have to squint. Okay, good. A dog is following the scent of a jackrabbit. Which of the following accurately describes how the dog's brain integrates information for smell? A, chemoreceptors in the brain send impulses to the smell in the nasal cavity. Okay, so as soon as we're dealing with receptors here, what type of, what chapter does this remind you of? You remember?
Where we talked about like the kinases and like all that stuff.
Yes,
that was very confusing.
There was a lot, yeah, cell signaling. So yeah. Cell signaling, with neurons, what do we remember happens? With neurons, we have chemoreceptors, right? We have chemoreceptors and we have those neurons with, and that's called synaptic signaling, right? It's a form of short distance signaling. And let's see, does A make sense? The brain sends impulses to the smell. That doesn't really make sense. Chemoreceptor cells in the nasal cavity send impulses to the appropriate area of the brain. That makes sense because, right? Because remember, it's short distance signaling. All these neurons are firing chemicals back and forth until they reach the brain. So this seems to be the potential answer. So I'm going to go with that for right now. Chemoreceptors on epithelial cells of the tongue send hormones to the appropriate area of the brain. We're not dealing with epithelial cells. We're dealing with neurons. And D, receptors originating in the nose send action potentials to the motor regions of the brain. No, motor regions would have to deal with movements. So I don't think well, it could have to do with that, but I'm not sure what an action potential is. So I would go with B, which is the correct answer.
Could you explain why it wouldn't be A?
Why wouldn't be A? Yeah, because the brain isn't sending impulses to the nose. The nose is receiving chemicals, and that's triggering stuff to the brain. Okay, for the rest, rather than me just going through them, I think it would be better if you guys tried to go through them, and then we can work through areas that are tricky and that we're struggling on. So number 2, who wants to try number 2?
So like, do you want us to answer and then put our answer in the chat, or like how do you want to do this?
Kind of maybe read through the question and kind of go through your line of reasoning, whatever you prefer, and then we can help each other if we get stuck.
Okay, I can do 2 then. Give me 1 second, actually.
Sure.
Okay, so thrips are insects that feed on rose pollen. Scientists noted that the thrips population increased in the spring and decreased dramatically during the summer. The researchers hypothesized that food abundance was the limiting factor for the population. Which of the following types of data would be most useful for the scientists to collect at regular intervals on a designated test plot of rose plants? Amount of sunlight, mean temperature, density of rose pollen, and amount of pollen produced by each flower.
So what do you think?
So first it says that thrips are insects that feed on rose pollen, so that's their food source, I'm assuming. So I think it would be C, because because it's a limiting factor for the population, so they would obviously focus on their food production.
You're absolutely right. It is C because yeah, exactly what you said. I don't need to restate what you said. I think you did it perfectly. Okay, number 3. Who wants to try number 3? I'm not going to pick on people. I don't want to do that, but I'll just, if no one else wants to do it, Mira, if you're okay with it, we can alternate. And if anyone else wants to join in at any time, you're free to do that, completely free to do that. Okay. Number 3, if ATP breakdown, which is hydrolysis, remember hydrolysis is breaking down using water, right? Remember, break down the words, hydrolysis, using water to lyse it, to break it down. Whereas condensation reactions or dehydration reactions, they take away water to condense 2 monomers into a polymer. Okay. If ATP breakdown or hydrolysis is inhibited, which of the following types of movement across cell membranes is also inhibited? Movement of oxygen into a cell? Well, we know that oxygen is a small non-polar molecule, so it can easily go through the cell membrane. It doesn't need energy to go through the cell, right? So it's not A. Movement of water through aquaporins? Well, we don't need energy for aquaporins because aquaporins are channels where facilitated diffusion happens, right? Because that's where it's passive transport. Passage of a solute against its concentration gradient? I think this is the answer because we need ATP to work for active transport. This is against its concentration gradient. Or D, facilitated diffusion. This is also passive, and this one does not require ATP. So the answer is C. And that's correct. Okay, good. And just a reminder, like, these are actually from the AP exam. So if you can answer these pretty well, then honestly, you're in pretty good shape for both the final and probably the AP exam as well. Okay. Go ahead.
Okay. Which of the following questions is most relevant to understanding the Calvin cycle? So the Calvin cycle is the light independent reaction, right?
Yes, no? It's light independent.
Okay.
Exactly.
All right. So I don't think it's A because it's independent from light. How is ATP used in the formation of 3-carbon carbohydrates? How is NADP plus reduced to NADPH? And how was ATP produced in chemiosmosis? I think it's B, but I have no idea. Like I honestly, this whole unit was kind of a bit confusing for me.
Would it be
Sure.
C?
I would think it's C, but let me check the answer real quick.
Yeah.
It's actually B. Oh, that's right. Duh. It is B. Sorry.
Like 3-carbon carbohydrates.
Yeah, I was
right?
getting it confused with Krebs cycle because in Krebs cycle, the NADH becomes important for ETC, but that's not right. We need to remember in photosynthesis, this happens in light-dependent reactions.
Yeah.
ATP is produced in chemiosmosis also happens in light-dependent. That's between photosystem 2 and photosystem 1. Remember, we have chemiosmosis happening.
Yeah.
Exactly. And NADP plus reduced to NADPH, that happens at the end of the light-dependent reactions. So when we get to Calvin's cycle, we're worried about creating G3P or PGAL, as some people call it, glyceraldehyde 3-phosphate. That's what we're concerned with.
So basically, you could use process of elimination for this?
Yes, exactly. Okay. All right. Next one. The equation above shows one of the reversible reactions that occur in the blood. Okay. So this is saying a proton or hydrogen ion plus hydrogen. This looks like it's carbonic acid, but it could be hydrogen carbonate, the gas. Wait, actually no. This is hydrogen carbonate. I think it's an ion. Okay, and that produces water and carbon dioxide. Okay. And this looks like it's an acid, honestly. But anyways, let's see. The equation above shows one of the reversible reactions. And athlete's blood pH, yeah, and that's why it's important, because we're dealing with pH here, that you recognize. You don't need to know that this is an acid, but knowing chemistry helps to know that this is an acid. After exercise, an athlete's blood pH has dropped below the normal level. Okay, pause real quick. What does it mean for pH to drop? pH to drop means acidity increased, and acidity increases means that there's more hydrogen protons, right? Less pH means more hydrogen protons. How will normal blood pH be restored? So Mira, this is an example of negative feedback because we're restoring something.
Got it, okay.
An increase in O2 concentration in the plasma will lead to an increase in H+ concentration.
But that would make it more acidic, right?
Yeah, that would make it more acidic. No. Obviously, this is wrong as well because that would make it more acidic. And an increase in sweating will lead to a decrease in OH- and H+ concentration. That doesn't quite make sense to me because it could be, right? Because hydroxide concentration has to do with bases, right? And if you decrease both of them, that's kind of not doing anything. Maybe it could be that, but I don't think it's C. An increase in breathing rate will lead to a decrease in blood CO2 and H+ concentration.
Yeah,
that seems right to me, right? Yeah, because also if this decreases, then as a result, the product produced will also decrease.
So more hydrogen ions means more acidity.
Yes, more hydrogen ions means more acidity. And if you want to remember it mathematically, pH is negative log hydrogen concentration. So the more H+ you have, the less the pH.
Gotcha,
okay. In 1944, Avery, MacLeod, and McCarty performed transformation experiments. We haven't learned about this yet, but we can try. Using live harmless bacteria and extracts from virulent bacteria treated with various enzymes. Oh, actually, I think this part we haven't learned about the transforming factor, but the part about enzymes we have. So we can definitely answer this question. Which of the following enzymes were used and why? Oh, Mira, do you want to go through this one?
Oh, this is not one of my strong units. You should go through this.
Okay. Proteases and RNases to rule out protein and RNA as transforming factors. Lipase, an enzyme that facilitates the breakdown of lipids to rule out lipoproteins as the transforming factor. Kinase, an enzyme that facilitates the transfer, blah, 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 to show that transformation is phosphorylation dependent. ATPase to show that transformation is not dependent on ATP. Well, the transforming factor is RNA or DNA, some sort of nucleic acid. So I would say A then, I would guess, because you would try to rule it out using, so you would use so whether it's protein or RNA, we don't know. So yeah, you kind of do need the background information about the experiment. But basically, the whole idea behind this is if a protein is breaking it down, if proteases are there, then and then the virus dies, then that means it's not the transforming. it has to do with control group. So essentially, we know that RNA is the transforming factor, right? Transforming factor. It's genetic material. Yeah, I just wanted to confirm that. Sorry. So, RNA, if RNase was put in there, then the transforming factor would be gone, and we would notice an effect. So RNase is our experimental group, I guess you could say, or positive control, as some people would say. And then protease is going to break down protein. And then if we see that nothing happened, that means protein isn't the transforming factor. Thus, protease would be our negative control group, or just our control group. Okay.
So basically, you could like rule out the other ones because it doesn't have anything to do with the DNA.
Yeah, it doesn't have anything to do with genetic material. Okay.
Okay, got it.
Okay. The process illustrated in the models depicted above all result in which of the following? So we learned about crossing over and fertilization. We haven't learned about conjugation, but I'll just give you a preview so you can answer this question. Conjugation is where bacteria, they use their pili. Remember pili? We did learn about pili. They connect their pili together, and these are called sex pili. So they connect their pili together, and they kind of switch over their genetic material. So this guy gives his genetic material to her. She gives, obviously they don't have genders, I'm just saying, just to get a visual. And then this person gives his genetic material over to the other lady or man, whatever it is. Okay. The processes illustrated depict which of the following? So is this transcription?
B, I think.
B, yeah, definitely. It's definitely, forgot to bold this one. Let me do that real quick. Okay, sorry, yes, it is B, an increase in genetic variation. We talked about earlier, Hiba, right, with meiosis, how crossing over increases genetic variation. Okay. All right, Mira, do you want to try the next one?
Yeah, sure. The vertebrate forelimb initially develops in the embryo as a solid mass of tissue. As development progresses, the solid mass near the end of the forelimb is remodeled into individual digits. Which of the following best explains the role of apoptosis in the remodeling of the forelimb?
Do you remember what apoptosis is?
The cell death, right?
Yeah,
no?
programmed cell death, yes.
Programmed cell death.
The opposite of apoptosis would be cancer, because that's uncontrolled cell division.
Got it, okay. Okay, so apoptosis replaces old cells with new ones that are less likely to contain mutations. Apoptosis involves the regulated activation of proteins in specific cells of the developing forelimb that leads to the death of those cells. Apoptosis involves the destruction of extra cells in the developing forelimb, and apoptosis in the developing forelimb triggers a differentiation. I think it's B, because it's a regulated activation, so right?
Yeah, I agree. B is the correct answer. Because you just need to know that apoptosis is programmed cell death, and it has to do with regulated activation. This was in our unit on cell signaling, because it's going to receive chemicals or some sort of hormone or maybe some sort of cell receptor that's going to bind onto a ligand on the cell. That's going to induce, through some sort of signal transduction cascade, it'll activate some proteins by phosphorylating them. Remember, with kinases activate proteins, and so on and so forth. Okay. Oh, a graph question. Fun. Okay. What most likely causes the trends in oxygen concentration shown in the graph? So this is in the water of a lake, and during darkness we can see that. So during the day it increases, and then during the night oxygen concentration decreases. So without looking at the answer choices, we can think about why potentially. This either deals with cell respiration or photosynthesis. I would think it has to do with photosynthesis because we're dealing with light and dark, right? So during the day, plants are creating oxygen and they're pushing that out through the stomata, right? Whereas during the night, they're also using up oxygen, and also other animals are using it. So I would say it has to do with, let's see.
It's not A, right? I think we could eliminate that one.
No, I don't think that makes sense. Respiration, most organisms, that could be the answer, but I think it has. This and this seem like it could be both. Like if you didn't know anything else, you would like these two are too similar. So if either of these was right, then it wouldn't really make sense logically. So I would go with D because it produces more oxygen that is consumed by respiration during the day. That makes more sense. Okay. And yes, the correct answer is D.
Because like everything, like during the darkness, everything's like asleep and like basically, right?
Yep. Okay. Oh, so it's not 47 questions. It's yeah. I was like, we're going awfully too fast for this to be 47 questions. Okay. A new mutation that arose. Oh, sorry, Mira, it's your turn.
Okay. A new mutation that arose in one copy of gene X in a somatic cell resulted in the formation of a tumor. Which of the following pieces of evidence best describes how the new mutation directly caused a tumor? Okay. So new mutation directly caused a tumor.
So tumor is what? Tumor has to do with, we talked about this earlier, not apoptosis, but?
Cancer?
Yep, cancer. Tumor has to do with cancer. And what is cancer?
Uncontrolled cell growth.
Yep, uncontrolled cell growth and division. So which of these answer choices?
So A?
Yes, it is
No.
A.
Yeah.
It is A.
Okay.
Because protein X, if it's mutated, it creates an overactive version and then it becomes uncontrolled, right? Okay. If it's decreasing the stability of protein X, potentially, but that doesn't really answer the question. Overactive version of protein X, but once again, that doesn't answer the question. If it blocks the cell cycle, then that's good, because what happens is actually in our cells, remember those checkpoints that we have in our cell cycle? What happens is it has to pass through each of those cells, those checkpoints. If it doesn't pass through those checkpoints, then it will just stop, and then potentially apoptosis will occur. Because you know what? Because the cell is willing to sacrifice itself, its life, to ensure the survival of the organism. Because if the cell, but then what happens is sometimes because of mutations, the cells get confused and they become selfish and greedy, and they're like, no, I want to keep growing and I want to keep dividing. So that's what happens. Okay. Okay. Diagram. Also fun. All right. Sorry, I just need to digest the diagram a little bit. So on the left, we have some mucus. We have a phospholipid bilayer. This is the inside of the cell, and this is the outside of the cell. It looks like in this CTFR protein, the chlorine ions are going against their concentration gradient. Doesn't it look like that to me? That's what it looks like to me. And then that's a normal. But in cystic fibrosis, when there's dehydrated mucus, it doesn't allow the chlorine ions to pass through. Okay, anyways.
Wait, so which one's the inside and which one's the outside again?
Cytosol is the inside, so the top is the inside, and extracellular.
Got it. Okay.
Cystic fibrosis is recessive. Blah blah blah. Okay, that's not important. Prevents normal movement. As a consequence, the mucus layer that is normally present on the surface becomes exceptionally dehydrated and viscous. So this becomes extra thick. An answer to which of the following questions would provide the most information about the association between the CFTR proteins and the viscous mucus? Is the mucus also secreted from the cells through the CFTR proteins? We're not really worried about mucus being secreted, so that doesn't make sense. How does the disrupted chloride movement affect the movement of sodium ions and water by the cell? That seems potentially it could be right. I don't know yet. How does the mutation affect the structure? It doesn't look like the structure of the protein necessarily is too deformed. It actually could be C, because we know that mutations in proteins lead to a different structure. So it could be C. And D, what is the change in the nucleotide sequence that results in the CFTR mutation?
Oh wait, I think it is C, because like based on its structure, that's what permits it to go through and not stay back, right?
I agree. I think it would be C. Let me look at the answer key. Oh, it's actually B.
Oh, wait.
Okay, let's think about this for a second. Let's try and explain this. So
I think maybe because the chlorine stops moving through, and that's what causes the mucus to get dehydrated.
Yeah, what Zach was saying, there's like proof of that in the diagram. So like obviously I think it has to be backed up, you know?
I see what you're saying. Yeah, because it has to, so Zachary's saying it has to do more with the chloride, the movement of chloride ions. Okay. So and then that would affect the water. Okay.
Oh, because it's saying the extracellular fluid, maybe that like has to do with the solvent. I'm not sure.
Yeah, because then honestly, C and D are too similar for it to be. Yeah.
Yeah. Because it has to do with the change in something, so makes sense.
Yeah. Okay. If chemical signals in the cytoplasm, oh, your turn.
If chemical signals in the cytoplasm control the progression of a cell to the M phase of the cell cycle, then fusion of a cell in G1 with the cell in the early M phase would most likely result in the...
That's a weird question.
I read it in the weird tone that I didn't. Okay. If chemical signals in the cytoplasm control the progression of a cell to the M phase of the cell cycle, then fusion of a cell in G1 with a cell in the early M phase would most likely result in the...
Chemical signals in the cytoplasm control this progression. Okay. Fusion of, so for some reason a G1 cell is fusing with a cell in early M phase. So that's like prophase probably. Like let's say prophase. So let's say this G1 cell is fusing with a cell in prophase. What would happen? Would the replication of chromosomes only in the G1 cell? It hasn't gone through the synthesis phase yet, so that wouldn't make sense. Exiting of both cells from the cell cycle and into the G0 phase? Maybe, but I don't know. Condensation of chromatin and preparation of nuclear division? That seems like the best answer, I think, because we're in prophase, right?
Yeah.
Okay, let's see in the chat. Did someone say something in the chat? No, the chat isn't popping up. When do you see the chat? Ah, there we go. Oh, yeah, no problem, Alan. Of course, and I'll post the rest. We'll post the rest. We're almost done, so we'll post it and then you can see it on YouTube. All right.
Thank you, MJ.
All right, bye. Okay, and then we have transfer of organelles from G1 cell to the cell in M phase. It wouldn't make sense. Why would the G1 transfer its organelles? I think it's C.
Oh, C? Yeah, that's what I think.
We're dealing with prophase. I think it's, yeah, I think what this question is testing you is whether you know what happens in prophase, and when prophase happens. And the correct answer is C. Okay, we're good. All right. So this actually is, this is transcription, which we haven't learned about yet, but I think the question has to do more with something else, something that we have learned. So, Mira, do you want to try?
Yeah, sure. The figure above depicts the DNA protein complex that is assembled at the transcriptional start site of gene X when the expression of gene X is activated in liver cells. Previous studies have shown that gene X is never expressed in nerve cells. Based on the diagram, which of the following most likely contributes to the specific expression pattern of gene X? So which contributes to the expression pattern of gene X? All right. I feel like A is too broad of an answer, but
I agree. A doesn't seem right.
General transcription. C also seems weird. I'm not sure.
C seems weird too. Yeah.
Some tissues, but not other tissues. That just throws me off. I feel like it has to do with DNA. So I don't know. I mean, B also has like RNA, which has to do with genetic material, but it's either B or D, I think. I'm not sure.
Well, maybe B, because it's blocking because the question says it's not active in.
I think, yeah, I think it's B. Let me see the answers. The answer is C. It's not B.
Oh.
The activator is a sequence. Yeah, okay. So the activator is, we haven't learned about this yet, but it has to do with transcription. But the activator is a protein. It's a protein that binds to DNA, and then it basically, that's what activates DNA transcription.
General transcription factors. Okay, I see.
Yeah, okay. This one we wouldn't know. That's fine. Next one. The figure above shows a model of a ligand precursor being cleaved. So this ligand precursor is being cleaved to produce an active ligand that binds to a specific receptor. So it binds to. So
does cleave mean like change shape or like remove, like alter its shape?
Cleavage.
Oh, okay.
And another vocabulary that they use a lot is change in conformation. Conformation means change in shape.
Shape.
Yes. Okay. Conformation is shape. Okay. Okay. Being produced to produce an active ligand that binds to a specific receptor, which of the following is most likely to reduce the binding of the active ligand to its receptor?
To reduce.
Reduce the binding of the ligand to its receptor. A change in the cytoskeletal attachment of transmembrane proteins. Maybe, but
it doesn't mention it, but
yeah.
I don't know. I think it's B, but
presence of a large amount of precursor for form of the ligand. So this would probably have to do with some sort of feedback mechanism. So
okay.
if you have a large amount of precursor form, then it wouldn't bind to the receptor. So it seems, that makes logical sense.
Right, because it changed the shape.
Yeah.
Kind of.
So then the ligand precursor, because the ligand precursor wouldn't fit into the active receptor.
Yeah.
Increasing the ratio of the number of unsaturated, no, that doesn't make sense.
No, it doesn't make sense.
A mutation in the receptor gene that causes substitution, no. Well, this could, a mutation in the receptor gene that causes a substitution of charged amino acid for nonpolar.
It doesn't talk about its shape, though. I mean,
well, this would inherently change its shape.
That would change the shape.
Because the mutation,
the amino acids.
Oh yeah.
It could be B or D.
I feel like D's more specific.
Because if it was B, if there's more precursor forms, then can't they just also go through cleavage and then
more? Yeah, you're right.
Because you can't really reverse the mutation.
So what would happen is it looks like enzymes actually. There's like, you can graphically measure it. As you have more and more ligand, it'll also connect to more and more receptors. But after a certain saturation point, then it levels off. So I was thinking of the leveling off, but the D makes more sense because that mutation would would permanently, like it would change the shape of the receptor and it would no longer bind. So I think that's the right answer. Let's see. Yes, that's the right answer. Good. Is this the last question? Yeah, we're on the last question. Okay. I'm just gonna hit enter a few times so it's all on one page. Or I could have hit page break. That would have probably been more efficient. But okay. All right. Do you want to try this one?
Yeah, sure. Students in the class measured the mass of various living organisms. They then kept the organisms in the dark for 24 hours before remeasuring them. None of the organisms were provided with nutrients during the 24-hour period. The data are as follows. Which of the following is the best explanation for the pattern of change in mass of the organisms over time? Okay, starting mass, final mass. Then they kept the organisms in the dark. Okay. Which
it looks like they all look like they lost mass, right?
Yeah. So
but not a lot, like only a little bit.
Yeah. So
well, it can't be C, the law of conservation of matter, because that would be, it's contrary to that. And D, growth and reproduction.
D is like, that's increasing.
That would be increasing. So it can't be those two. I would think it's A, but
A, right?
And cell respiration have to do with this, potentially?
With loss of something? I feel like as they lose, they gain. So I don't feel like it would be B. I feel like it's A because it's water loss.
It might, because it says that they're not provided with nutrients, right? So if they're losing their sugar, like that's what.
Then they can't really do cellular respiration, can they? I'm not sure.
Well, they already have some, so they're like, obviously it's not going to be like as much cellular respiration, but I mean, if you stop eating for a day, your body doesn't stop to like cellular respirate. So
because
water loss due to evaporation. Well, they're all in the water.
Like, it's because they're in the dark. So if they're in the dark, then they can't do evaporate. Like
Yeah, how would they
Evaporation can't
evaporate?
happen.
Yeah, how would that happen? So how would cell respiration lead?
Yeah, I mean,
to
I feel like
decreasing
it's not
mass?
specific enough, you know, like the answer they give. I feel like there has to be more than just stating
It's like how
cellular
your body
respiration.
burns calories, like just by you, like you
I
don't
see
have
what
to,
you're saying.
like just by living,
Mm-hmm.
you know, you like burn calories, and even if you
Mm-hmm.
don't eat, you will continue to burn calories.
I see what you're saying. So you're saying that the glucose is being broken down into ATP and that ATP is being used, essentially.
Yeah,
And
because
that's
they
leading
still
to,
need
okay.
ATP to survive, and they have stored, still have stored energy that
Yeah.
they're using because they're not receiving
Yeah,
nutrients.
cellular respiration makes sense more than water loss. I feel like at this point.
And yes, the correct answer is cell respiration. Okay, good.
Yeah.
All right, I think we did pretty decently. I don't think we did. I know there were some that were. These are pretty hard. I mean, honestly, these are. Some of these were easier than others. But...
they're easier than Miss Rouse test, I think. Feel like because there's more context, right?
Mm-hmm. Yeah,
so
there's a little bit more.
Room.
Yeah. Okay. There's. I'll just post the answer key because I think that'll probably be easier. And there's. Chandana wrote explanations for some of them. I don't think she was able to do it for all of them, but that's fine. The rest, of course, people can watch the video. I'll post it in the description box in the video itself on YouTube. Okay? Do you guys have any final questions?
Quick question, not about the content, but how are you studying?
How am I studying?
Yeah.
I mean, I'm kind of just going through practice questions. That's probably the best way for me to study at this point because for me, I think staring at a textbook doesn't really help and going through notes. For me, I think the more questions I do, I can figure out, okay, this is where potentially and then I can just, it's a quick reminder of what it is that I need to remember. And also B, if I don't know something, it becomes very apparent very quickly that I don't know this. Let me take a quick look at it. And I think that's more effective for me, at least.
Gotcha.
Okay. All right. I'm going to stop the recording. As always, you can always go back to the recording and watch it later if you need to. And please follow Revise Now on social media. I know most of you, I think all of you here are probably, so that's good. But if you're watching this video at a later time, please do that. We're on Instagram and Facebook, and please subscribe to our YouTube channel. That would be great. All right. Bye.